to our Thursday research conference, which, as you know, we have continued into the virtual era, although we missed the fun of being in the Vic Theater. So it gives me a great honor to introduce a longtime friend and colleague of mine, Dr. Steve Labkoff, who who is a physician by training. He trained in cardiology. He also trained in informatics at the same place I did a number of years ago. I think he came a year or two after me. We both trained under Bob Greenis at Brigham and Women's Hospital in Boston in informatics, which is part of the Harvard medical complex. Steve has followed an industry path. He's worked for a number of companies over the years, a couple of pharmaceutical companies. He's also been involved in a lot of entrepreneurial activities, and so brings a nice industry-focused perspective to his talk. Most recently, and now, he is Chief Data Officer for the Multiple Myeloma Research Foundation, and he leads their bioinformatics department that's responsible for architecture, execution, and management of all of their data assets. And he is going to talk to us today about the work that he does, and will also give us some insights into his industry perspective on informatics. And so I will turn things over to Steve. You should be able to take the screen and show your slides.
Yep, 1 second. Let's hopefully this will work. We tested it beforehand.
Yep.
Let me just put this on macro view.
Great.
Hopefully this is sharing properly. See the
Yeah.
screen?
This looks fine to me.
Thank you, Bill. Yeah, Bill and I were in a different era at this point. Bill was in the early 90s, I was in the mid 90s. And I actually wasn't even clear I could make a living at informatics when I started. It was a random encounter with one of Bill's colleagues, Jim McClay, who actually got me into informatics at an AMIA meeting back in 1992. And if it wasn't for a random chance meeting of Jim McClay and him giving me his business card, I never would have met Bob and eventually Bill and everybody else in the field. I'm going to talk to you today about some work I'm doing in the multiple myeloma space. As Bill indicated, I'm a cardiologist by training. I'm not a hematologist or an oncologist, but this work revolves around a lot of big data questions and problems, and we've really encountered a ton of them along the way. And I'm going to talk to you first about the introduction to what we've done, and then next I'm going to go through the registry and how it works, and then the challenges that we've hit. And the challenges are really varied. They run the gamut from typical informatics issues to legal to regulatory issues to some issues around how people actually pack and address boxes, and I'll get into all those things. So who is the MMRF? The MMRF is a research foundation that's been around for the last 22 years. It was started by a woman named Kathy Giusti. Kathy is currently our chief mission officer. She herself is a myeloma survivor. Back in the day when she was first diagnosed, she was fortunate enough to have a twin sister and access to some early drugs in the myeloma space. And she, unlike most others, survived. And when she got past her disease or got through it, she decided to start this research foundation to try to answer questions that really weren't being answered in the typical academic setting. So the MMRF works a little bit more like a business than it does an academic institution, although it does have very strong ties into academia. We have three main goals at the moment, and they're revolving around immuno-oncology and genomics. A second one is, how do you support an organization like this through venture? And so the MMRF actually has started and is running its own venture capital fund, the profits from which are pooled back into the MMRF to help sustain it. And then we also have a third pillar, which is the one I'm involved in, which is democratizing data so that patients can understand and leverage it for their own purposes, for their own therapy and their own disease course. We work off of a strategic plan. This was a project that was put together with some help with folks from IQVIA. They helped us actually build a formal plan like we would have built when I was at Pfizer or at AstraZeneca. And those three pillars are part of it. We have a host of advisors from all over the country, in some cases the world, and they really run the gamut from academia through industry. MMRF actually can proudly say that it's been behind, or at least in part responsible for bringing more than 11 new drugs to market. And when I say in part, it has either provided data or grants or other types of work in kind to help advance these drugs. It currently is focused in on various clinical trials that it itself sponsors and others that it supports in multiple different domains, including antibodies, BiTEs, and CAR T therapy. So as the CureCloud as the MMRF matured, it started to realize that it was really, really good at generating novel data sets. And early on, it was the organization that actually caused the myeloma genome to be sequenced. Well, this is going back almost 15 years. And because of that, that was one of the first cancers to actually be decoded. And that data was then used by life science companies to actually help them looking for new compounds and bringing those to market. Eventually, it evolved to do one of the longest studies that's out there in myeloma called the COMPASS trial. The COMPASS trial is ongoing. It's got 1,154 patients in it. Unfortunately, not all of them are still alive. Only about 640 are still with us. And it's been running since 2011. That trial has basically been following clinical data of these patients as well as their genomic data for the last 9 years. And it's scheduled to come to a natural conclusion in 2023. But before it does, we're going to be doing immunoprofiling on all the patients within the COMPASS study. And I'll talk about that in some detail a little bit later because it involves what we're doing in CureCloud. Eventually, they put together a few other studies at University of Michigan where they actually got another 1,000 patients and their specimens genomically sequenced. And then that led to another clinical trial, which is not represented here, called My Drug, which is testing out genomic variants in various clinical trial arms. So My Drug is a platform trial, and it has sort of works like various cassettes. You can drop in a cassette for a specific drug with inclusion and exclusion criteria, and it uses all of the existing infrastructure for the trial, so you can bring a new, we call them cassettes, into the program as you go. And this is one of the first trials looking at genomic sequencing as genomic aberrations as an inclusion or exclusion criteria for these arms. So that's an ongoing trial. Work there actually led to what I'm working on, which is called the CureCloud, which is a direct-to-patient registry, which is being built and stood up to actually bring in data around 5,000 patients for myeloma. And I'll go into all those details as we proceed. So what actually is the CureCloud? Well, its overreaching vision is to provide the most comprehensive data resource for patients, clinicians, and researchers concerning the diagnosis and treatment of myeloma, and to facilitate a cure for every patient. So that's a wonderful high 50,000-foot view, but let's unpack it a little bit. So what is it actually? What are we trying to do from a goals and objectives perspective? We are trying to recruit 5,000 patients with active disease, 4,500 of which will have active myeloma, 500 of which will have smoldering myeloma. For those of you who are clinicians, you may recall there's various different phases of the disease and its lifespan. We start off with MGUS, which progresses in a handful of cases to smoldering myeloma, which eventually goes to full-blown multiple myeloma. And then you enter into the realm of the disease and its recurrences and all of the therapies needed there. We are planning on collecting and returning clinical genomic data to patients through means of a 70-gene myeloma-specific panel through blood biopsies, which is a fancy word for a tube of blood. That tube of blood is a special Streck tube, which is actually able to isolate free circulating DNA. That DNA is expanded through PCR, and it's run against the 70 gene panel. We are also saving 3 vials of blood from the assay for future work, including immunoprofiling. And on top of all of that, we're getting broad consent from the patients to actually abstract out all of their clinical data from their EHR where it's relevant to myeloma. And we're building a clinical repository of patients in a longitudinal fashion. Every 6 months, we're actually querying their electronic health records and bringing more of that data to bear to build this out for the long haul. Using that 70 gene panel, we're going to be matching patients to clinical trials that have a genomic component to them. Right now, there aren't all that many. There's about 6 or 8 of them. But if the patients have the right genomic sequence, or not sequence, or the right genomic makeup, they will be given sort of a direction to hit up the PIs and try to get themselves enrolled. We're also creating a data set that will support research and development as well as health outcomes research within life science organizations. And this is actually a really crucial piece of the program, because it's through this piece of the program we hope to be able to sort of self-fund what we're doing. This is a $40 million venture, and it is currently 100% funded through donations from patients. Some of those patients happen to be very wealthy, thank goodness, and their donations have brought us roughly half of what we need. But in order to sustain this for the next 10 years, we do need to find ways to leverage the data assets to help keep this thing going. And at the end of all this, we're going to be building a data set to provide both patients, clinicians, and researchers a very high quality data set for their research and for their clinical care. All this is expected to run a minimum of 10 years and is been my every waking moment for the last 2 years. Let's see here. So what's the registry? Mechanically, if you double click on it yet again, it's a massive data collection workflow management engine. The data repository is, as I said, meant to collect information for these patients. And then we have portals that are either up or being built that will provide access to the constituencies for their use. For patients, we have visualizations, data visualizations. We have access to patient support documentation. We have a clinical nurses that actually call the patients every 3 months and take notes on them and where they are in their disease, and we keep that in a separate portion of the CureCloud. For clinicians, we provide them with a clinical genomic report that's read out by the pathologists at the Dana-Farber Cancer Institute. And for researchers, the research database will be made available for use once we hit approximately 500 patients that have walked through the entirety of all the workflows. We actually need to bring in at least 7 different types of actually matched data. And the matched piece is actually quite important. In many cases, when you try to aggregate data across multiple data sets for patients, it's not always easy to get exactly the same patient's data, and you need to resort to things like probabilistic matching. In the case of the CureCloud, with one exception, all the data will be actually matched. The one exception is we're going to cross that bridge sometime at the end of next year, which is medical claims from payers. And we're hoping to get matched data there. We're unclear if we'll be successful just based upon how they make data like that available. So as you click down even further, this is sort of a high-level workflow of what we've built. Basically, a patient comes to our front door, they sign up, they run through inclusion-exclusion criteria. If they meet inclusion criteria, they sign a very deep patient consent that is broad consent. They answer a survey, which gives us some initial demographics and some information about their journey to date. Then we have a phlebotomy company that sets up a blood draw, and this is a level of complexity that is part of the program. They send the phlebotomist to the patient's homes. And I'll explain why we do that in just a second. Because in COVID, that's turned out to be actually a really good thing. But we didn't know it at the time when it was planned. We also, when the consent is signed, we send off the patient's name and the consent and HIPAA consents off to a third party who then does EHR data abstraction for us. The phlebotomy company goes off. They draw 4 tubes of blood. One goes off to the Broad, where the 70-gene panel is run, and the other 3 go to the Mayo Clinic's biobank in Arizona, where they sit in for us to use at a later date. When the 70-gene panel is actually completed, all of the BAM files and VCF files and all the accompanying files that go with that get transferred to the Dana-Farber, where we have clinical pathologists who actually read all these files looking for the calls, the variant calls in the panel. They sign off on an actual genomic report, which is sent back to us. We send that report off to the clinicians. And most importantly, we send the report off to a genomic counseling organization, which digests the report into 8th grade English. And I mean that quite sincerely. It's written at the 8th grade reading level, and that report is returned to patients. So they're able to make use of it and understand the depths of what all this means. And that report is sent back to the patient. All of this is kept inside of the CureCloud data system, which is actually a distributed set of databases that are linked. We have the EHR data, we have all the clinical genomic data, we have the patient reports, the genomic reports, and those in gray are planned for next year and the year after, which will be patient-reported outcomes, immune profiling, claims, and of course our clinical nurses' notes, but those will never be made available until we can come up with a means of absolute de-identification. We have all of this has been 2 years, or actually more than 2 years, but since I got here, it's been the last 2 years of my life building this with a number of partners. And we stood this up and opened the front door on this on July 14th of 2020 inside the COVID pandemic. And I'll tell you about all the challenges that have happened there, not the least of which was our phlebotomy company went Chapter 11 3 days before we opened, and we had to sort of jump through a bunch of hoops to replace them in the month of July. You might ask yourself, why did we pick 5,000 patients? Well, when you start, one of the main findings from Compass was that there were 11 or 12 genomic variants or genomic findings or subgroups or subtypes that were part of the Compass data set. And you'll notice that I have here various weightings in terms of how frequently they turned up within Compass in the really ugly lime green color in the second column. We also wanted to break this down according to where they were in their journey. So we have newly diagnosed relapse 1, 2, and 3+. If you break those down according to the percentages and according to the N of what we expect to see, we wanted to see if we could get patient numbers and cohorts that were at least 40 for study. And if you do that at 1,000, most of this heat map here turns out to be red. Even at 2,500, there's still a lot of orange and red, more green. But when you hit 5,000, the model turns mostly green. Now, this isn't the only way to slice and dice the patients. It's not the only way to slice and dice these cohorts, but it is one that from our Compass work seemed to be one of high importance. And this was one of the driving factors in getting us to these numbers. So we're not doing this in a vacuum. We're doing this alongside a number of academic institutional advisors. We have an old friend of myself and of Bill's, Charlie Safran's one of our advisors. We have folks from ASH, from MSK, Weill Cornell, and a bunch of others here who are academic advisors. And they have different positions that involve Odyssey and other relevant research efforts. We also have a life science advisory board from companies that actually have an interest in myeloma who provide us with input in terms of what their long-term needs are going to be in terms of how we build out the data set and how we provision it for their use. We're trying very hard to work towards common data standards. One of my previous jobs, I worked at Intelligent Medical Objects, a company that Bill's aware of, and I'm sure many of you have heard of. I have a very soft spot for using all standardized vocabularies and ontologies. We're actually participants on OMOP for oncology, which is part of the Odyssey program. We're trying to help bring some standards into that area. We've examined EMCODE. I'm not clear we're going to go with EMCODE or code for EMCODE just yet, It's an ASCO standard that's trying to evolve. Unfortunately for us, it's more of a solid tumor standard for data nomenclature and ontology. It's probably not one we'll take on just yet. We are working with ASH to try to make our registry, and they have a myeloma registry as well. We're trying to make it so that users of each could actually crosswalk back to the other through an anonymized token mechanism so that we can complement each other's work. So we did open for research mode in the fall of 2019. We ran about 200 patients through that. And we have, but the research mode wasn't doing one of the real core missions for the program, which was to return data to patients. For that, we needed to get our program CLIA certified. It needed to be CLIA certified, and the entire pipeline had to go through CLIA certification. And that didn't get done by the end of 2019. But we did open for research, where we collected the blood, we did the phlebotomy, we started EHR data abstractions, and we ran the panel in research mode. As I said, we ran about 200 patients through that, and then when we opened in July, this slide's actually a little outdated now, it's October, we now have close to 300 patients who have signed up and have accrued as requesting to be participating in the program. I didn't expect us to recruit this fast. We're recruiting at roughly, depending on how you count, either twice our expected rate or 4 times our expected rate, since we have a very complex consent process that requires a physician to order the test and then to sign off on it. We have over close to 600 patients who have signed up, but not all their physicians have signed the paperwork yet. So we're working on getting that going. So we're close to 600 who are part of the study at this point since July 14th, which to my mind is more than remarkable. So where are we in terms of the our roadmap? Again, we opened in 2019. Over the course of 2020, we opened in July. We look to be bringing in PROs and full exome sequences, as well as immune profiling, and then later in 22 or before 22, if we're lucky, we'll bring in claims data. I mentioned earlier that we'll have 500 patients with smoldering disease. That's going to be the source of our exome sequences, our next-gen exomes, whole exome sequences. And we're going to be following those patients especially closely because of those 500, some of them are going to be converting into myeloma, and we'll catch their journey. Unlike with Compass, we'll catch those patients from the very, very beginning of their journey. And there's no reason we couldn't actually increase these recruitments other than we would just need more money. So this is the roadmap of where we're going. At opening, we've had 4 data sets that are part of the system. The patient-donated data that's part of our demographic survey at the beginning, our nursing notes, the EHR data abstractions, which are building a 24-table relational database that's updated every 6 months, and as well as the 70-gene panel with the BAM math files and all the somatic mutation data that's coming out of that. So we're about halfway through the talk, and I want to tell you that this has been an absolutely staggering amount of work to happen. And throughout all this, it sounds, maybe to you may sound like this was easy. It was anything but easy. We hit roadblocks in more places than I could possibly have ever guessed. I've done very complex programs in my career, one of which was actually building a hospital in Kampala, Uganda once for Pfizer. And I thought that was the most complicated thing I'd ever done, involving traveling 8,000 miles and working with the CDC and procuring satellites and satellite dishes and time and raising capital to do it. It turned out that putting that hospital together was just a cakewalk in comparison to this program. And that's because this program involved an enormous amount of regulatory and legal issues, the issues around clinical data reuse and abstraction. The returning data to patients became a tremendous source of complexity for this program, and we'll go through all these in the next few slides. So one of our macro challenges was basically building this registry for all comers. So typically registries are built by an academic institution or perhaps a pharmaceutical company. They tend to work within the confines of of one institution or group of institutions. The first macro challenge here is that we're trying to get data from patients across the US, and as long as you met the inclusion criteria, you could participate, which means that we had to figure out a way to get electronic health records data out of EHRs across the US. And as much as I'm a fan of FHIR and I'm a fan of being able to make those kind of digital connections, the reality of it is that in 2020, 2018 through 2020, most of those connections, except for a handful of large institutions, simply aren't ready for prime time, especially for types of data we're trying to collect. Some of the specialty data, like fish reports, are coming in as faxes or as PDF images, and you just simply can't get them through the kind of interfaces that we were hoping for. The home-based phlebotomy program became a real challenge for us. Things as simple as getting the kits shipped from the Broad, having them labeled properly, and having them tagged right involve manual workflow. And as soon as you introduce a human being into a workflow like this, any number of weird things can start to happen. And we've seen pretty much all the weird things that we think can happen, but we're only into it now, again, for like 200 that we've actually run. And even in there, we're seeing a 3 to 5%, I'll call it a weird factor, of weird things that just happen. So an example of a weird thing that happens is that we have some exclusion states. We can't run this test in the state of New York, in Alaska or Hawaii. Alaska and Hawaii are simply based on practical issues around FedEx shipping. We have to get the blood into the Broad inside of 24 hours so it can be fractionated while it's still good. So Alaska and Hawaii, FedEx could not guarantee us next day delivery. So we nixed them on that basis. New York, on the other hand, we had to nix because it turns out when you're trying to get CLIA certification, New York has its own super CLIA certification. New York State has its own review process for getting labs certified for patients who live in the state of New York. And it turns out because this was a brand new assay, getting that certification was not feasible at the Broad in time for this to get off the ground. So therefore, unfortunately for us, we're not able to take anybody from the state of New York. So among the weird things that have happened, we've had patients who want to participate who live in New York but have homes in Florida. So what have they done? They have literally set up, even in the pandemic, to try to coordinate when our kits would be shipped and the blood would be drawn so they would be in their Florida homes in order to participate in the CureCloud. Technically, they can do that as long as they have a clinical physician who's not a New York doctor who can write the order in Florida. That's kosher. But anything in the state of New York is not kosher for the time being. We're going to work on solving that one, but that was a bridge too far to get open. Other problems that we'll get into a little more deeply had to do with moving genomic files between institutions, between the Broad and the Dana-Farber. That involved using systems from Google and a homegrown system from the Broad called Terra that has to then communicate with a commercial system that the Dana-Farber uses in their pathology department to bring in those files and then make those calls by the pathologist. That became another one of our challenges. And then creation and agreement on various data dictionaries and data models, and to try to use common standards where possible. So we've been exploring, as I mentioned earlier, the OMOP Common Data Model for Oncology, which, as much as we want to use it, some aspects of OMOP for Oncology aren't ready for prime time either. So we've dug in, rolled up our sleeves, and we have members of my team who are working to actually help bring some of those standards to bear so that model can be evolved. We had technical challenges with all the various data handoffs. We had to ensure that confidentiality and privacy was of prime importance. In the background of this whole thing, we have a Rosetta Stone that basically aligns all of the various identifiers for the patients in the system. That Rosetta Stone is encrypted and behind firewalls, and every one of our partnering organizations has its own series of unique identifiers for patients within the system. It's only inside the system where the patients can be reconnected. We had to put together sound data stewardship principles and execute on those principles. End user, as an example, our end users who are administrators, we don't just let people like email in and sign up. We physically have to lay eyes on them and know them personally before they're given access credentials to administer anything within our system. So even if you were to hack in with credentials, if you hadn't been known to us, you wouldn't be able to just get in. Workflow administration and data flow monitoring is also a really key part of this. We had to build a back-end administrative portal for this so we could actually watch all of these different pieces of the system and where people's data were along the way. So I talked about weird things happening. So one phlebotomist, we have two packages that go out. The package goes out with tubes that have to be drawn and sent to the biobank and tubes that have to be sent to the Broad Institute. Well, naturally, even though we have instructions and labels in two separate boxes, the tubes got confused. And so far, even in the real version, the CLIA version, that happened twice out of the first 24 kits that we sent out. So we have to accommodate, issues like that and send out fresh kits and things along those lines. And I talked about common data models. From a legal and regulatory perspective, getting this CLIA pipeline certified was really critical. In order to return data to patients, that's why we had to go with a CLIA certified test. Well, having the CLIA certified test actually has some knock-on things we had to address. So in order to get a CLIA test ordered, it has to be actually ordered by a physician. Well, this had another knock-on effect, which was as we started digging on this, it turns out that you needed to have physicians in the same state as the patient in roughly 18 states in the union. In 15 states, it's actually illegal for a doctor across state lines to write the order for the blood test in a patient in another state. In 3 states, it's ambiguous and leaning towards illegal. These laws turn out to be part of the telemedicine laws that until COVID were pretty restrictive. And as a result, this took us some time to figure out what to do. Our solution for this ended up being that we could have done several things, one of which could have been hire doctors in all 50 states to be our physician writers. But we thought the better of that, and we actually have the patient's personal clinicians, their hematologists, writing the order. That way, we can return the clinical report back to that physician, and it can help inform them for their patient's actual clinical care. So that their clinician gets the report, the patient gets the digested report, and the two of them, we're hoping it'll facilitate a better conversation about their entry into clinical trials or any changes in their therapy based upon the data that we have here. Another complexity was dealing with not one, not two, but count them, 3 IRBs. Because we work with the MMRF deals with Western IRB, which is a paid IRB or commercial IRB. But because this program is being read out and it's still a clinical study and the Dana-Farber is involved, we had to deal with their IRB. And again, same thing with the Broad. Broad has their own IRB. So various stages of the game here, the protocols and various aspects of the project had to go through all these different external checks and balances, which is not a bad thing. It just added to complexity. I mentioned New York being problematic. And then we have another issue which came to light, which was how we get the medical records pulled. So it turns out that there's a e-consent law that was put on the books in 2001, but not all the hospitals across the US have heard of that particular law. And as a result, they're refusing to honor the patient's signed digital consent roughly 20% of the time. And that's another regulatory and legal hurdle we're trying to cross by strengthening our digital consent and getting certified signatures and things like that. So that was another series of confounders that we had to deal with. Yet others were around data reusability and the abstraction, or data reuse and abstraction. So again, we went through all comers. So that meant, and we tried to go with FHIR, we tried to figure out a way to build pipes into all these different places. But since we don't know a priori where these patients are coming from, they could be coming from a doctor's office all the way through to the Mayo Clinic or Cleveland Clinic or Harvard. Building all the pipelines at the moment is still a kind of prohibitive exercise for us. As a result, we hired a third party who does this. They're actually getting back, wait for it, PDF files. When they go and make these requests, and when they are getting them back, they're getting PDF files of the clinical record, which then have to be physically manually reviewed and manually abstract it for all the data that we're looking for. That, in fact, is about a third of the expense of this program. And that's, we're plowing through that one with brute force. And I know there's folks on the line who are going to say, we should be able to do this digitally. And I would wholeheartedly agree. And if you could show me a way to do it across all comers in a consistent fashion, please contact me after this talk. I am all ears. We would love to figure that piece out and stop with the manual abstraction. It's a high expense and it's a very complex thing to have to go through. We do have data dictionaries in the back of all this. This is just a screenshot of one of them. We define in very low detail all the different values and what the control values need to be. We've got them all set up in a way that the data as it comes in could be put into things like R, so we can do our own research and visualizations on them. When it comes down to returning the data to patients, I think I've already mentioned this had to be a CLIA certified test, but also making sure that all the data was sent around through the different parts of the pipeline in an anonymized fashion required us to hook up different connection points between large institutions. And hooking up these types of pipes between places like Harvard, you'd think that since the Broad's a Harvard institution and so is the Dana-Farber, that wouldn't have been easy. Well, that was a good 6-month exercise by itself. We had to build new API calls and build them from scratch to make sure that all the data was being sent across properly. And again, I mentioned earlier, we had to monitor all the data flow to ensure that we're not losing records along the way, especially some of these large records. Something as trivial as building a clinical report, the physical report that people are going to have to read, that had to be crafted from scratch. The Dana-Farber pathologists and the MMRF spent almost a year, believe it or not, crafting what would physically go into the report that the clinicians would get. And of course, that had to go for IRB review and had to go in for the CLIA certification. And all the report automation pieces had to be built from scratch as well. And then finding a way to return the report back to the clinicians of record, that had to be built. And as much as I would love to send this all back through, an encoded email, some docs are not giving us their email addresses. So we have to resort to, wait for it, faxes. And we had to accommodate faxes in this workflow in order to make sure we could return data to patients appropriately. This is just a copy or screenshots of the clinical report from the Broad and the Dana-Farber. And on the other side of the screen is the MyGene Council report. This is the 8th grade reading level report that is going back to the patients. Returning the data to patients was yet another wonderful exercise. Visualizing data, highly complex data that is usable by patients, again, at the 8th grade reading level, we had to bring in a separate firm whose job it was to ensure that all of the visualizations were easy to understand and very easy to digest by real people, as opposed to clinicians or PhDs. They went through rounds and rounds and rounds of A/B testing with both clinicians and with patients to try to hone in on what would be the most advantageous and easily digested approach to showing patients this data. And that's still, frankly, a work in progress. That's not even complete yet. The next couple screens are just a couple of examples of some of these, the screenshots of the visualizations that are in flight. We're trying to make big fonts and with large iconography to try and make this as human digestible as possible. This one on the far lower right, we're trying to actually also do patient journey on some of the temporal data. So we can actually show when labs change over time. We have the lab data, we have their fish data, we have all the abstracted information. So we can show this over time and give them a view as to how their disease is progressing, at least as we're seeing it through the CHR abstraction process. We have more views of that information. Security and confidentiality is obviously an enormous chunk of work on this program. Everything that we do is SSL encrypted. We have end-to-end encryption. Data is encrypted at motion and at rest. All the identifiers are blinded except for the data managers here at the MMRF and our a vendor who's working with us to build the backend systems. The Rosetta Stone is completely encrypted, and we have role-based security, and every single person involved here has to have hand-approved access to the system. We had to establish a project management office. You know, the last time I dealt with a project management office of this complexity was again in Africa, and this was even more complex. We had to, when I joined the organization, we had 14 different companies that the MMRF had contracted with to help build this thing. We winnowed that down to 8, but even with 8, that's still a lot of people and programs and processes to manage. It's actually enormous. We had to do edge case generation and with multiple whiteboard sessions to figure out where different issues were going to be hit for exclusion or for edge cases for when something were to happen. So when the phlebotomist puts the label on the wrong kit, we had to think about that and come up with a way to nullify that kit and resend a new kit and take them out, reset the system. So every single little nit that was possible to happen, we tried to pre-think and come up with all these and come up with ways to deal with all these different processes through the system. We built dozens, if not 100 different data flow diagrams of how things are going. This is just one example. This particular example is just the first of 9 on the data flows and approvals necessary. And this is just to get a patient into the system. And these all link off to other sheets and so forth. And then, of course, the biggest challenge of all, which was like a bus hitting us from left field, was COVID. When COVID happened in March, all the work that was in flight at the Broad came to a screeching halt. We couldn't finish the validation of the CLIA test. It all literally stopped because the Broad was conscripted by the state of Massachusetts to do all COVID testing all the time for about 5 months. They only recently came out of that and have been back to doing our work, our work and other work in the research space, probably since only September. Likewise, Dana-Farber closed for all research programs. No patients were being accrued for any study, and our work was put on the back burner for about 3 or 4 months at the Dana-Farber. As I mentioned earlier, our home phlebotomy company went Chapter 11 due to COVID on July 11th, which required a rapid RFP team to get into position to try to bring a new company online. We actually pulled that off. We got a subsidiary of Quest Diagnostics to come to the table, and they're now our phlebotomy company. Overall, COVID set us back probably more like 4 months. But again, the first blood kits went out on the 14th of September, and our kits were returned. The first 12 hit the analyzers about 2.5 weeks ago, and we're expecting next week to get the first genomic tests off the analyzers and over to the pathologists to start actually closing the loop all the way through and returning this data to patients. As we go here, our program will have 4 types of data as we open. I've mentioned the patient-donated data, the nurses' notes from they call the patients every 3 months to see how they're doing. I think I forgot to mention, if the patients have a relapse, actually, we're doing another blood draw. So we sort of hit a big green button or a big red button, depending on your perspective, and that sets off another blood draw for those patients so we can serially follow their somatic mutations. Because in this situation, unlike a germline mutation, the patient's mutations tend to change with therapy. And as a result, if you catch them at different stages of their disease before or after a given therapy line, you probably we think we're going to see changes in their actual somatic mutations. So that's why we're going to the trouble of actually getting that repeated. We have the EHR data, which I've already mentioned, and then our data from our genomic panel. In the years to come, we're going to be adding patient-reported outcomes. We're looking to put in the aortic QLQ30 and the MY20 as patient-reported outcomes. We hope to be bringing that online in early 21. New profiling will be a complementary series of tests with CyTOF and single-cell RNA-seq information that will be on every patient in the CureCloud. We'll have whole exome sequencing on the patients who are in for smoldering, medical claims later, probably realistically by 2022. So, and we opened the door. This is the front page of the system. I'm not going to do a system demo today, but if you go to mmrfcurecloud.org, you too could do this. And only if you're a member, if you have the inclusion criteria, but you could go and take a look at what we put together, and it is up and working. I'll just add one thing. When I joined this organization, to me, this was, the informatics program that would just be like this amazingly complex thing to solve. And I love solving these types of puzzles. But this program got very, very personal to me in March when my first cousin was unfortunately diagnosed with myeloma himself. And from that point, he just had his transplant about 3 weeks ago. So this program has gotten exceptionally personal for me. It's not often that you work on something like this and it hits home in this kind of way, but it really has come home in a painful way to me in that front. So I'm going to pause here and open up the conversation for questions. Again, if anybody, by the way, has any ideas about anything I've talked about or concepts or things that they think could help us with this, I'm going to put my email address and phone number in the chat box. That's my email address, and my cell is, I'll give you my my cell. If anybody has any suggestions for anything I've talked through here today, we are not proud. We will take any advice we can get. We are open to hearing, anything that would help us make this easier. Because as you've seen, this has been, an adventure in complexity. So I'll pause there and I'll turn it back over to Bill to moderate questions and answers.
Yeah, great. Thanks, Steve. Unfortunately, we can't really clap. Well, I suppose we could clap virtually. No, it's a very nice talk to hear. Always good for me to find out what my friends are up to, and it sounds like you've been up to a lot. I'm going to make 2 quick observations and then a question. I see some accumulating in the chat box, and you can answer those as well, or people can unmute themselves. A couple of comments. One is that one of the nice things about your talk, as I think we have students who are very deep into bioinformatics and very deep into clinical informatics. And your talk and what you're doing is a great example of how those areas overlap. And I think as time goes on, we're going to see more of that. A second observation is that your story of all the challenges you faced remind us of that old adage that when it comes to informatics, technology is the easy part. A lot of times it's dealing with complexities with organizations and people and so forth. And I think you drive that home, even though the technology is very interesting. Let me ask a quick question. I don't want to take up all your question time, but it's interesting that you're starting to import EHR data, and obviously that's important, although there's a number of people listening here, myself included, who have spent time kind of thinking about what we can actually do with electronic health record data. Sometimes it's incomplete because patients get their care in different places. Sometimes it is inaccurate. Clinicians don't track things right. And so sometimes, actually having people extract or annotate the data can actually overcome some of those problems. What do you hope to do in the longer run with EHR data to help inform your project?
So that's a great question. So one of the things we've done, and it's into another program I'm running for the for MMRF, it's a program with the Intermountain Health Organization. We actually have built a clinical data visualizer to actually look at longitudinal healthcare data using Sankey plots as one example, where we actually can take what we want to see. We want to map this to outcomes. So what we really want here is the clinical information linked to outcomes. So then we can show, in the case of drug regimens, as an example, and I don't have it in this deck to share, but what you can do is you can actually track the course of their drug therapy and their outcomes and what different clinical states they proceeded through. So this will be familiar to you, Bill. I actually been working with Bob Greenis on work that we did way back at the Brigham on clinical state mapping. So the clinical state mapping work that Greenis worked on back in the 90s, we brought to bear for this, and we're hoping to be able to identify clinical states along their journey where you could have a different intervention that might actually lead to a better outcome. So it's another piece of the visualization that's meant for the clinicians. It's part of a different program, but that's one example of where we're hoping to go with this data. You said that bringing the clinical data forward could, it's full of holes, it's got issues. It absolutely does. And by the way, one of the vendors that we worked with, I won't say who it was, but as we were bringing, we had more than one vendor involved with the clinical data piece of this at the beginning. And one of these vendors actually, I found an aberration in their data, which was they had start dates in one table, but no end dates for most of the drug regimens that were being used. But yet in another table, they had duration of therapy. So I called them on that and I said, how do you calculate duration of therapy if you have no end dates back in the other table, which was in theory what they were abstracting into. And it turned out, and I still to this day can't believe it, but they were imputing end dates based upon what was in textbook information and putting that in the data set, which made my mind want to explode. And we actually canceled the contract. It was a $5 million contract, and it was canceled. Because I don't know how many different ways you can say you can't do that, but you can't do that. And that was,
great. So yeah, there's a few questions in the chat box. I don't know if you can see them or not. I can read them to you if not. Jennifer Sullivan, one of our PhD students, thank you for sharing your experience. How did you learn about the various regulatory requirements for everything you wanted to do? Whom did you consult, or what resources did you use?
Yeah, so the MMRF has a lot of organizations that has partnerships with, many of whom are actually law firms. And one of those is a law firm called Ropes and Gray, a regulatory firm out of Washington, D.C. And as you know, I've been around the informatics world and in the business world for an awful long time. And some of this stuff, I just started to sort of ask questions. And when we started asking the questions, it became, well, we don't actually know. Then we went to Ropes and Gray, who then found all these things out. Like I would never have figured out that the telemedicine laws were the laws that governed a project like this, but they are. And Ropes and Gray gave us the whole point of view that it was the 18 states that you couldn't do this in the way we had envisioned. We envisioned having our chief medical officer write all the orders, and that would have ended it all. It would have been much simpler. But they told us, you just can't do that. Now, post-COVID, it's going to be very interesting to see if some of those laws are sufficiently relaxed so that we could go back and do that again. But I think where we landed with having the clinician of record write the order is probably a really good solution, because it actually helps bring the doctor and the patient together. And I think that's all it's said and done, that's a really good thing.
Yeah, I agree with you. I think the telemedicine laws are going to change. I mean, telemedicine's had a huge impact at our institution for a while, over half of our visits. So there's another question from Chris Alpe, who's the director of our library, and I'll read it. What method did you use to negotiate the disagreements on data visualizations? Would you recommend doing all the multiple rounds of A/B testing on variations? How many rounds did you do? I don't know if you got all that.
I got it. I think she meant agreements, not disagreements. Oh, maybe she meant disagreements.
Yeah, probably meant to.
It's either or. How did you get to agreement?
So I won't lie to you. I think disagreements actually is the right word, because we had some very heated discussions over the year in terms of what to show and what not to show. And I didn't run that particular part of the program. I had to delegate a lot of things in here. You can't imagine this would be run by one person. We have a cast of hundreds, maybe not hundreds, dozens working on this program. But we had an external group come in. Their names the group is called Springbox, and they actually they did a lot of work in the space of patient-facing information. And they are the ones who ran the A/B testing. Honestly, I can't tell you how many rounds they ran, but they did run several rounds of A/B testing on various, like every tile in the visualizations that were in there, they were pretty much all tested. But I didn't run the piece of the project, and I wish I could tell you it was, 6 rounds, but I simply don't know.
Thank you.
Sure.
Yeah, great.
Anything
Any
else?
other questions? If not, I'll actually ask one more. You didn't say anything about, or if you did, I apologize if I missed it, about clinical trials. Is this resource amenable to clinical trials?
So this is a clinical trial. I think I mentioned that. This has an NCTID number. We are in clinicaltrials.gov, and one of the outputs on the patient report, which is let's see if I can get back there. There it is. On the patient report, one of the outcomes of that report is a clinical trial matching service. So there's really we find at the moment there's about 8 clinical trials, I think 6 to 8 clinical trials that have output from CureCloud that would be used as a inclusion criteria for that trial. And if the patient meets those criteria, they are shunted or at least given the advice to contact the PI to get into that trial. One of those trials is one of our trials. It's the one I mentioned earlier, the My Drug trial. So, maybe it's a little self-serving, but we are trying to find patients for that trial too.
Okay, yeah, and I know there's at our Cancer Institute here, there's probably others who know more than I do, but there is a kind of precision medicine trial that is basically comparing people using molecular approaches versus not using them.
Right. And
okay. Any other questions or comments? Okay, well, seeing none, I want to thank Steve for sharing this very interesting project with us, and maybe down the road as it matures and achieves more things, we can get him to come back and give us an update. So thank you very much, Steve, and we appreciate you taking your time today to do this.
Thanks, Bill. Thanks, everybody. Have a nice day.
Yeah, you too.